Professor Noam Shomron. Noam heads the Genetic Intelligence Research Laboratory in the Faculty of Medicine here at the Tel Aviv University. He leads a multidisciplinary team of scientists, biologists, physicians, engineers, bioinformaticians, and mathematicians that aim to deepen the understanding of development of disease in order to generate significant impact through translational ideas. You don't put periods in your sentences.
I wanted it to be brief.
Brief. And his talk right now is going to be on big data and genomics, halting the spread of breast cancer.
Okay, good morning. I want to thank the organizers for inviting me to speak in this forum and in this session. I'm going to talk about big data and genomics, how we sequence a lot of information using advanced technologies, and about a specific case or story from our lab where we stop the spread of breast cancer. So this is the human DNA, at least a small part of it, represented by 4 letters A, G, T, and C. In 2001, when the first human DNA was sequenced, it cost the equivalent of 4 jumbo jet airplanes to sequence one human DNA from beginning to end. And today, it costs around a bicycle. So that means it's much cheaper, it's much more accessible, and anybody could read their own DNA. And of course, we have to understand what to do with all this information. Reading a lot of DNA can help us in a field called precision medicine. And what we define in precision medicine is reading the DNA of one individual, comparing it to another individual, and then trying to fit a treatment or a therapy or trying to fit a particular drug based on these DNA differences. Not necessarily 2 individuals, it could be a lot of information. And in our lab, we try to combine fields in order to bring the field closer to precision medicine. We don't only look at DNA, we look at DNA and RNA. We look at specific tissue and surrogate tissue. You can't always interrogate the tissue you're interested in. And there's a lot of big data out there, a lot of information. And we're trying to retrieve from this big data, a handful of genes that are relevant. It could be 1, 2, 3 genes, but the big data doesn't help us. It helps us to filter and to find the genes that might cause a disease. So this is what we do in the lab in terms of precision medicine, and I want to show you how we performed several of our experiments. This is the central dogma of gene expression. 1 unit, a gene is transcribed and then translated into a protein. That was up till about 15 years ago. In the past 15 years, we know that a lot of RNA is expressed in our cell. It's stuck at the RNA level. And some of it binds our other RNA and shuts off its expression. And we use deep sequencing and NGS technologies in order to enter the cell and to read all the RNA in it. And once we read all the RNA, we see that a lot of RNA is expressed from the DNA, up to 70%. However, only up to 3% of that RNA is translated to a protein. So it's either a terrible evolutionary mistake of our cell, or it means something, and we're trying to understand it. So in the past few years in our lab, we sequenced about 1,500 individuals, and we built tools to try and filter this information, to understand it, and to look for causative mutations. We and many others focus on the coding regions because we understand them better, and we understand what happens when we find a mutation there. So this is an overview of what we do in our analysis. You can see from left to right, we're looking at the severity of the mutations. You can see inside exons, missense mutations, and nonsense mutations. That's the moving through the circles from left to right. And if we look from right to left, we're looking at the prevalence of these mutations in the population. So less than 5% allele frequency, less than 1%, and then novel. So in the center, when we sequence an individual, we try to find mutations that might cause to a disease by changing the structure of a protein. It's easier for us to look at proteins again because we can understand what a mutation does there. In a study we performed together with Professor Nadir Arber and Professor Eitan Friedman, we also scanned and looked at asymptomatic individuals. So 25 healthy individuals enter the clinic at Professor Nadir Arber's lab in Ichilov, And they defined actionable mutations. So by looking at their DNA, they decided what might have implications for changing your lifestyle or maybe increased screening. You can see the 25 individuals on the x-axis here, 1 to 25, and the number of actionable variants on the y-axis. You can see that we found actionable variants in all of these patients, divided into 3 groups: cancer, cardiovascular, and pharmacogenomics. Now, it's not really a surprise because the genes we looked at are common genes that are also screened without this exome analysis or whole genome analysis. There's BRCA, there's CYP genes, there are other types of genes there. But this was a comprehensive study of reading an entire DNA from beginning to end. Now, we know the sequencing is very easy to do now. And, we're getting used to interpretation, and we're fine with interpretation. But the big task here was for the physician to sit in front of their patient and convey this information. Which information are they going to tell them, and what are they going to do with this data later on? So we wanted to show that not only the coding genes are relevant for detrimental effects on an organ or on a human body, it's also worth looking at the non-coding regions that could have a major effect on disease development. So we took this approach. We took large data sets, 3 of them, and we combined them together. The first data set is the entire mutations of a person. It could also be a data set of mutations. And it could also be SNPs. SNPs are single nucleotide polymorphisms, which mean that these are known mutations in the population which are prevalent more than 1%. So the first database is a very large list of mutations or polymorphisms. The other set are genes of interest. So these are oncogenes in this case here. The third set are regulatory sites. These are called microRNA binding sites. Now let's imagine a cell. And if you enter the cell has a lot of mRNA in it. And in order to shut off the expression, a microRNA would bind to it and would shut off its expression. Now, we can identify these interactions between a microRNA and an mRNA. And we know where these regulatory sites land. So if we look at the intersection of these 3 sets, in the center, we will get changes in the population that happen to land in a regulatory site, that happen to land in genes of interest. In this case, these are oncogenes. So we can separate the population into those genes that could be regulated or couldn't be regulated by a regulator called microRNAs. This was the general scheme, and now I'm going to talk specifically about the study we performed, which also involved nanoparticles at the advanced stages of the experiments. We decided to look at breast cancer due to the prevalence in the population. 1 in 8 women will be diagnosed with breast cancer throughout their lifetime. The other reason is that early detection is becoming more common due to education, and obviously the earlier you detect, the better the outcome of the patient. And then we took these data sets and we crossed them, just like I mentioned before in the overview. So these are now real numbers of our experiments. We took breast cancer-related genes, any of those associated with breast cancer in the past. So we received about 4,000 genes here. These are too many to look at, obviously. So we crossed them with those that have a microRNA binding site, that a microRNA could bind to it and regulate it. We removed about half of them here. That's still a lot of genes to look at. And then we crossed them with those that have a polymorphic site, or known to have different nucleotides in the binding site of these genes related to breast cancer. These are still quite a lot, about 190. And the final cross-section we performed is those genes that are related to cytoskeleton organization. Now, because we wanted to target metastatic breast cancer, we wanted to target those that will affect the movement of the cell. In order for a cell, a cancer cell, to move from the primary site to the secondary site, it has to really change its code and its structure. It changes its cytoskeleton, then it passes into the bloodstream and then squeezes again at a new site. Now, we thought that if we could get it stuck really open in the primary site, it won't be able to move to the secondary site. Therefore, we performed this last filter looking at cytoskeleton organization genes. Now we received 19 genes. So 19 genes is a reasonable amount of genes to look at. Half of them were related to cancer. 1/3 of them are related to movement of cancer from one side to the other. But we picked one which is less known, called Paladin. There's about 10 to 20 papers about Paladin. And we took it as a case study to show that one nucleotide change in this gene can potentially affect the outcome of the disease. So the first assays we did, this is the Paladin gene. It's associated with the cytoskeleton organization with structures called podosomes. It's the finger-like structures at the edge of the cells that allow the movement and the adhesion of the cells. The first experiments we performed were on the cancer features of Paladin and these microRNAs in cell lines. So here you can see different assays we performed on cancer features. And what we did is we injected or transfected the two microRNAs, miR182 and miR96, which regulate this gene, Paladin, in order to see that when we increase the microRNAs, Paladin goes down and there's less movement. You can see that on the left-hand side in the scratch assay. You can also see it in the invasion and migration. So we can elevate these regulators. We can downregulate the Paladin gene, which is involved in cytoskeleton organization, and we can affect cancer features in vitro in cell lines that we grow in a plate. Okay, so this is in vitro. This is very nice. What happens in an animal? The next experiment, we generated these breast cancer cell lines with a red color in them, so we can follow them using microscopy. And we performed this experiment that you can see on top. We allowed breast cancer tumors to develop to about 2 weeks, and the size was quite large, about 0.5 centimeter. Then we excised them out of the mice, and we allowed another 20 days for these mice to grow in their cages. Now, we developed 3 different cell lines. One of them is the cancer cell line with a scrambled small RNA. This was our control. Another one was a cell line that expressed really highly miR182, and the third one was with miR96. So potentially elevated microRNAs will slow down the movement of these cells. I want to remind you that we didn't want to target the primary tumor. Here you can see using the Maestro microscope that we didn't target the primary tumor. The primary tumor is fine. It's the same size. We wanted to target the metastases. Here I'm showing you again the values of the primary tumor on the top left-hand side. We didn't affect the primary tumor. We wanted to affect the movement of the cells. And at the bottom you can see the weight of the animals that didn't change in the various experiments. So 20 days after tumor removal, with the 3 different cell lines, we took these animals, we performed a CT imaging, and we looked at them in the Maestro microscope in order to look at the metastases at various sites. These cells are red. And we also sent it to pathology. I'm showing you the lungs of these experiments. On the top, in the control, using the control cells, you can see a halo of red colors in all the lungs. And in the other experiments, in the overexpressing 182 and 96, except for one artifact in the center, we didn't see any halo, we didn't see any movement of the cells from the breast cancer, primary breast cancer, to the lungs. And you can see the quantitation on the right-hand side. So at this stage, you can say, okay, this is a little bit cheating, because a cancer cell never arrives together with its drug into the animal or into the person. So can we deliver it from the outside and show that it's effective as well? So this was our third and last experiment. As you can see, the scheme was similar on top with one small change. So we had 2 weeks of cancer initiation, removal of the primary tumor, and then 20 days later, we took the animals to evaluate whether there's any metastatic sites. However, 2 days before removal of the tumor, we injected a hydrogel. So we contacted a colleague at MIT, Dr. Natalie Arzi, who developed these hydrogel with nanoparticles in them that secreted our 182, our miR96, and a control sample. You can see a scheme of the nanoparticles here. Gold nanoparticles with fits where we could follow the green color inside the cells as well, with 2 different microRNAs in a control, each one on different beads, and also addition of cisplatin. Cisplatin is a drug that's sometimes used for treating breast cancer. It could treat the primary tumor, but our target was also the secondary tumor. We wanted to show that there could be some combinatorial effect here. 2 days before the operation, the removal of the primary tumor, these animals will receive this hydrogel, in the microenvironment of the tumor, a few injections in the microenvironment of the tumor. During these 48 hours, the nanoparticles released our microRNAs, potentially entering these tumors and potentially affecting the genes that we want them to affect. So these are the animals here. You can see this is without the drug, without the cisplatin. You can see the tumor in red. You can see the nanoparticles in green. And you can see we did not affect the primary tumor. And now you can see the effect together with the drug, together with cisplatin. You can see we have an effect also on the primary tumor, but our goal was a secondary tumor. 20 days after this experiment, we took these animals into the CT, we performed a maestro analysis, and we also sent them for pathology. Now I'm showing you the CT image on the bottom left-hand side. This is of our control group. Everywhere there's a red arrow that indicates a metastatic site in the lungs derived from the primary tumor that was initiated in the breast. You can see many tumors there. And now I'm going to increase the left-hand side of this image. And again, you can see the control groups. And you'll see very soon above it the images of our experiments. But before I tell you about the images or I show you the images of the experiments, I wanted to tell you that this is more than just an image. This is a beautiful image. This is the exact flash moment where everything came together. Years of work, over the weekend experiments repeated over and over and over again with tiny modifications every time. If you ever performed experiments, you probably feel my pain. This is the moment that every scientist lives for. So we have stopped the spread of breast cancer from the primary tumor to the secondary tumor in the lungs in most cases. 0 to 1 metastatic sites in the lungs using these gold nanoparticles with hydrogel, secreting 48 hours around the tumor and then probably affecting the genes of interest. This was proved later on, and downregulating these genes that allow the movement of these cells. It wasn't present in the lung, in the liver, in the brain, and other sites we removed from these animals after sacrification. In order to apply what we saw in animals into humans, we downloaded large databases, the TCGA databases, and we wanted to see whether there's a hint of anti-correlation between the microRNAs we looked at and other genes of interest, and also paladin. So here, at the bottom center, first of all, you can see the correlation between the two microRNAs. Both of them are highly expressed. They probably affect at the same time. And you can also see a very mild yet significant anti-correlation between the microRNA and the gene. You can see when one is up, the other is down. It's very mild, but it's very significant because there are many samples here. This is inside the tumor. This doesn't prove that it regulates it, but this is guilty by association. They are anti-correlative inside the tumor. In breast cancer, we also looked at other tumors, and we saw that it's applicable in other types of cancer as well. Another thing we looked at is the levels of paladin and lymph node metastatic sites. So can the metastatic primary tumor move on to adjacent organs? We saw that the higher the paladin, the more the cells move to adjacent organs. Again, this is not a direct proof, but this is guilty by association, and now we're investigating this further. So we believe we have a model now. This was only one gene, one proof that one nucleotide change in a non-coding region in the 3' UTR can have a drastic effect on the outcome of disease, at least here in breast cancer in mice. Now we have a model, so we cross these large data sets of known polymorphisms or known mutations and genes of interest, and we're looking at microRNA binding sites. We're picking these genes. First, we're testing them in our cells, making sure that there's a regulation between the microRNA and its target. The third thing is looking at large databases and looking in patients to see that there's a hint for an anti-correlation between a microRNA and its gene. And eventually, the last step would be testing it in animals as well, using these gold nanoparticles and hydrogels injected a few days before removal of the primary tumor. So in order to summarize what I talked about today, crossing large data sets can lead to specific genes of interest. Combining DNA and RNA can lead to key regulators in cancer. And precision medicine might be effective in breast cancer, at least in these models that we looked at in mice. Now, we believe we're looking at different aspects of precision medicine. We're not only looking at the DNA, we're looking at the changes in the DNA and how the RNA could be modified in order to regulate this DNA. We're looking at big data, but eventually, we come out with a handful of genes. And regarding specific tissue and surrogate tissue, we're not sampling the tumor samples any longer. What we're doing in mice is we're taking the blood from these mice, and we're using next-generation sequencing in order to sequence the small molecules inside the blood and try to identify the tumor and its mutations just by taking a blood sample. This is the wonderful team that performed the experiments, in particular the first 4 students that are written out here, and I thank you for your attention.
So we have a few minutes for questions.